Alright guys, so we are back. Just a few details missing. We'll fill those in and then we'll continue. Right, so I think the only thing I've forgotten is the hydrogen bonds. I'll do them in red because that's the colour pen I have in my hand. So 3, these are being broken, and 2. Okay, so we're looking at the function of DNA now, and one of its functions is it's got a structure which lends itself very well to being copied almost exactly. Okay, so it's got a structure that lends itself to that, and we're going to look now at how that happens. We discussed that because it's a very long molecule, because its hydrogen bonds can be broken easily, we said that that was good because we could replicate the molecule that way, and now we're going to see how. Okay? So what we're going to have to imagine is, first of all, what is causing this molecule to be ripped apart. And what we'll imagine is that there is a protein, an enzyme, that is moving along this molecule, and its only job is to separate the two strands, uncoil it first of all, and then break the hydrogen bonds. Now the enzyme that will be doing that as it goes along, okay, and we'll just have to imagine that it's there, is helicase. Okay? Helicase. So helicase is moving along, and as it moves along, it's separating these bases, breaking the hydrogen bonds. Very important, breaking the hydrogen bonds. Okay, so now what we have are exposed bases, right? Exposed bases, because helicase has gone down, separated the two strands, separated the bases from each other, they're no longer paired, they are exposed on two strands. Now remember, the cell has got, it puts a lot of energy into making sure that there are plenty of nucleotides around for when the replication needs to happen, or for when transcription needs to happen. Now, yeah, so what happens is this, and maybe I'll draw the new strand with a red pen. So what's going to happen is, now we've got exposed bases, all right? So these are all bases which need a base pairing partner, right? They've been separated from their partner by helicase, and now what's going to happen is you're going to have free nucleotides which will align to the bases that have been exposed. So we've got an A here, and if there's a free nucleotide with a T, or a thymine nitrogenous base, well, it will then align itself in the way that we've already discussed. Okay, phosphate group. There, so that would be T, and that would be its two hydrogen bonds between them. And over here, we will get, absolutely right, G, and you'll get that nucleotide lining up there. So free nucleotides align with their complementary base pairing partner because of hydrogen bonding. Okay, and this will happen here also. Here we'll have, nope, C. Right? Like so, and so on. Okay? So, then what needs to happen, so we've got the bases all lined up. If you can see, we're already starting to form our new DNA duplex right there, and the same thing will be happening on the other strand. Okay? But it's a little bit more complicated, and I don't want to go into the details. But just like what's happening on this strand, you're getting the same thing on the other strand. Okay, so we've got our new strand, new nucleotides lining up there, and you will notice they're exactly the same ones that were there on the other side before. So this A was base pairing with this T, and so we've got T here. This C was base pairing with this G, and we've got a G here. So the new strand is an exact copy. What we're going to end up is going to be an exact duplicate of the original molecule. Okay? Alright, so I forgot that one there. 3 there. Okay, 2 there. And then all that needs to happen is we need to join the nucleotides up to form the backbone. So if there's a fourth nucleotide there, it will be joined via the ester bond there. This one will be joined to that one. This one will be joined to that one. And suddenly we have a new DNA strand forming. The enzyme that does that, in this case, because we are joining DNA nucleotides together into a polymer, DNA polymerase. So two enzymes very important here. Helicase that breaks the hydrogen bonds, DNA polymerase, which then, once the nucleotides have aligned themselves with their complementary base pairing partner, DNA polymerase then joining those nucleotides up to make them into a polymer, into a strand, into the complementary strand. Okay? And then if this carries on the other one, what we'll have, if I denote the old strand, the parent strand with black, the new strands will be forming on the other side. And helicase will then carry on further down. We'll separate more of it, and we'll get more duplicated. Helicase will move further down, separate the rest of the molecule, and we'll then get two molecules where we had one parent strand, daughter strand. Okay guys, quite beautiful, quite amazing. I don't think I need to say more. Okay, so that's DNA replication. We'll now look at a slightly more interesting protein synthesis, which starts off with a very similar process. Okay, see you later. Right guys, so now we come to the, I would consider, the very important function of DNA, which is to encode proteins. Okay, now you've got to follow this very carefully. There's plenty of really excellent animations online. I'll do my best to explain it, but I would really suggest you have a look at those animations. I'm going to go through and tell you what you need to know, but do kind of look at what this might look like in kind of three-dimensional terms, or in real life. So let's begin. What's our starting material is, the DNA in the nucleus of the cell, right? So that's our nucleus. We have our nuclear pores. Okay. Now, what we eventually want to get to is a protein. So maybe this protein is, for example, an enzyme. Okay? Right? Now, here's the thing. This is nucleic acid. This is protein made of amino acids. It's folded up in a particular way. How do we get from there to there? Okay? Let's just do some brief labeling. That's our nucleus. nuclear pore, and finally this is the cytoplasm out here. Okay. Let's just see if that's all on the screen. Yep. Right, so, this is, the scene is set now. We need to get from the information that's in DNA, how does it give rise to a protein, a polypeptide chain, which is properly folded in 3 dimensions in the way that it is. All right? So, first of all, you've got to remember that this is a DNA double helix right there, it's condensed. But if we want this protein, we need to copy some of the information that's in the DNA, right? Now, why do we need to copy it? Because the DNA molecule, first of all, it's too big to come out of the nucleus, okay? And there is no second of all. It's too big to come out of the nucleus, so we need to have some way to get that information out of there into here. I should mention that, if you've seen my video on protein structure, we have discussed that this protein is folded in 3 dimensions in the way that it is because it's got all the amino acids in the polypeptide chain in the right order, okay? And what made all the amino acids in here in the right order, what determined that sequence of amino acid, what determines the primary structure, is the order of nitrogenous bases in the DNA. Okay? So we need to use that information of nitrogenous base sequence to make sure that we put the right amino acids in the right sequence for this protein. That information is there. The ribosomes, which are going to join our amino acids together, are out here. Okay, so the ribosomes are out here. Okay, so we've got a problem. Information is there, protein making machinery is out here. So we need to get the information out of there, out here. That DNA molecule is too big to come out, so what we'll do is we'll copy it. Do we need to copy all of it? No. We just need to copy the bit of the DNA that gives us the instructions to make that protein, all right? So, your DNA, all the DNA that's in your nucleus, there's only segments of it which makes up a gene, right? And that gene is the instructions for each individual protein, and you've got thousands of genes, i.e. you've got thousands of little bits or segments, lengths of that gene or of that DNA that gives you the instructions to make a protein. So, for this particular protein, let's just say it's the enzyme catalase, right? Now please don't shoot me if catalase has got a quaternary structure. We'll just call this catalase for example. Say that's catalase. Where's the gene for catalase? Oh, it's maybe that short segment right there, okay? So what we're going to do is we're going to copy that using the base pairing rules, all right? So just like, we looked at in DNA replication, the 2 strands were separated, the hydrogen bonds were broken, nucleotides came in, 3 nucleotides came in and aligned by the base pairing rules. A very similar procedure will happen, the only difference being that in this case it's going to be RNA nucleotides that are going to be coming in and lining up. Okay? The other difference being we're not copying the whole segment, we're just copying a short segment. And once our copying is complete, the DNA double helix is just going to rewind back up again into the double helix. And the other thing is that we're going to make a single stranded copy, and also we are going to have not T, but instead of it, we're going to have a uracil. Okay, so wherever we would have had a T nucleotide or a T nitrogenous base, it's going to be uracil instead. Okay, these are the key things that we're going to remember. So a bit of this double helix will unwind. Okay, hope you can see that. Maybe I'll just do it bigger. Okay, so say that's a bit of my double helix. Carries on, carries on. Right, now what's going to happen is this part encodes my gene for catalase, my enzyme. So what's going to happen is the double helix is going to... Actually, should I just draw the double helix? Let's just say that it is a double helix right there. Okay, so that's my double helix. Now what's going to happen is, if this is my section of DNA that I need to give me the information for the catalase, it's going to unwind. So instead of a double helix, it's going to start looking like that, okay? Two strands have come apart from each other, right? And we'll just kind of do a little example. Say the DNA sequence is G, G, GC, C, T, A. Okay? Now, when RNA polymerase comes along, and the RNA nucleotides are going to be lining up, which nucleotides are going to line up? We're going to have G to C, C to G, C to G, T to A to T, no, A to U, okay, U, and so on. And this process will carry on until we've copied the whole sequence that we need to give us the information to make the whole of the polypeptide chain for catalase. This is just a bit of it. Okay? So that process will carry on, and then the RNA will come off, and the DNA will rewind again. So once that has happened, we will have our DNA wound back up again, and we'll have our mRNA right next to it. But this is a short nucleic acid, it's a short polynucleotide, and it's small, and it can easily exit from the nucleus into the cytoplasm through the nuclear pore. Okay, and then it will be out here in the cytoplasm, where a ribosome may attach to it and start reading it. So first we must label this clearly. This is the messenger RNA, for the simple reason that it's taking a message from the nucleus into the cytoplasm, and the process that we've just described here is called transcription. Okay? We are essentially copying the information, and we're not changing the language that we're using to copy. It's still the language of nitrogenous bases. Okay? So transcription. Producing an mRNA which can move out the nuclear pore into the cytoplasm. Okay. Now the ribosome comes in. Now remember, the ribosome is made of ribosomal RNA. So, you know, it's a common question, what are the roles of RNA in protein synthesis? Well, you've got messenger RNA, you've got ribosomal RNA, and there'll also be transfer RNA, which we'll discuss later. Okay, so the ribosome is something that is made up of ribosomal RNA, or rRNA. Okay, now what will happen is that this mRNA, or the ribosome, will lock on to the mRNA and start reading the sequence of bases on the mRNA 3 by 3. We're just going to zoom into that process, or that process, and see what happens. So, for example, I can't even remember what sequence we used now. Let me just remember what it was that we used. So our mRNA sequence started off with C, G, G, and A, U, U. And, you know, this is just for example purposes, but it would also carry on for quite a long time. Okay, now what would happen is that, let's just get some terminology out of the way. This is called a codon. A codon, all right? And a codon is a very specific set of instructions for one amino acid, okay? So CGG will stand, or will give the instructions for a particular amino acid, and we'll see how that is. And AUU will also itself be an instruction for a different amino acid. So if you carry this sequence on along the whole mRNA, okay, so codon after codon after codon, 3 bases after 3 bases after 3 bases after 3 bases, if you carry this on, you will then get the instructions for a sequence of amino acids, because each set of 3 bases copied from DNA is the instructions for a particular amino acid, one of the 20. Okay? And so on. So how does this information get used to produce a polypeptide chain that can fold into 3 dimensions to make a functioning protein? Like so. So what the ribosome does is it provides an environment to join the mRNA and the tRNA. Okay? So let's just draw our ribosome there. Okay? So this is the codon, and the other player, our third important RNA molecule, is the transfer RNA. Okay? And I'll just make it look like a T, but it doesn't. And very importantly, our transfer RNA has got 2 important parts. One is an amino acid joined at the other end, and the other is 3 bases at its other end called an anticodon. Okay? Like so. Okay? Now, it's the tRNA that is the kind of translator. Okay? So now we are actually talking about the process called translation. We are converting a sequence of bases, or information in the form of a sequence of bases, into a sequence of amino acids. How is this done? So our tRNA contains has an amino acid at one end and an anticodon at the other end. Okay? And that any tRNA with the anticodon of GCC will always have GCC. No, sorry. Yeah, we'll always have arginine as its amino acid. Okay, so one of these 20 amino acids, any tRNA that can bind this codon through having a complementary anticodon will always have arginine at that end. Okay, and this is why the system works. So this tRNA will then be able to come in here. Its anticodon will pair up through complementary base pairing G, C, C. Okay, and its amino acid will be there. Okay, now what will happen is everything moves along one step. Right, so the ribosome moves along, or the mRNA is pulled through, however you want to look at it. Okay, so now the next one is AUU, and there will be lots of tRNAs, and whichever tRNA has the right anticodon to bind AUU will come here. Right, now this one will be UAA. So that tRNA's anticodon will be UAA, and that AUU, and any tRNA that can bind the AUU will always have at its other end the amino acid isoleucine. Okay, and what happens is when these two amino acids are next to each other, condensation reaction forms, condensation reaction occurs, peptide bond forms, and we have these two amino acids next to each other. Okay? And if this process carries on, so another tRNA will bind whatever that codon is, and according to that codon, a very particular amino acid will be brought in by that transfer RNA, and the third amino acid will be joined, and the fourth, and the fifth, and the sixth, and the seventh. Remember, the ribosome will be moving along to make this happen, but eventually we will get a long polypeptide chain of many amino acids joined together. The order of that amino acids was due to the sequence of codons on the mRNA, which determined which transfer RNA would come along and bind and bring up a specific amino acid. But the sequence of codons in the mRNA was dependent on the sequence of bases in the DNA. Okay? And once you have a polypeptide chain, it will then fold into secondary structure, tertiary structure, if it needs a separate polypeptide chain, quaternary structure, but eventually, because all the amino acids are in the right place, because all the R groups can form the right interactions, because they're in the right place, you will get a protein which is a three-dimensionally folded chain, properly folded so that it can perform its function. All because of the sequence of nitrogenous bases in the DNA molecule. Tell me something which is more fantastic than this. Exactly. Right, good luck guys. Any questions, put them down there and I'll get back to you as soon as I can, okay?